So let me welcome everyone to our today's theoretical and computational biophysics seminar. So it's a great pleasure to host Professor Jerome Baudry, an Illinois alumnus, actually, Beckman alumnus, I should say, today for our seminar. Before I introduce Jerome, a close colleague and a close friend, actually, so I would like to remind everyone to please turn off your microphone during the seminar. And if possible, keep your questions to the end where you can actually ask your question yourself. I'm gonna sort of give speaking privilege to everyone. Unless there is really pressing question, in which case I hope Jerome doesn't mind to be interrupted once or twice, maybe maximum during the talk. So that way we can keep it interactive. You can keep your cameras on. That gives a more interactive feel of the seminar. So that's great. Thanks, JC. But otherwise, turn off your microphones. So with that, it's a great pleasure to introduce Jerome today. So Jerome was trained in molecular biophysics initially by Jeremy Smith in Paris, where he got his PhD. Jeremy Smith was actually on my thesis defense committee also. That was a very nice kind of coincidence. But then after doing his PhD, he moves to Urbana-Champaign, Jerome, to do a postdoc with Klaus Schulten. And that was the time, 1998, 99, 2000. That was the period that when I visited actually Klaus Schulten for the first time from Germany. And Jerome and Ferenc, another close friend, graciously hosted me and took care of me when I was actually visiting Beckman Institute, which was a big place coming from Germany. So really, we accomplished a lot of work together back then, and we published actually even a couple of papers during those short visits. So after that, Jerome has had a detour to industry for a couple of years, where he was exposed more maybe to pharmaceutical applications of computation and simulation and modeling. And then he came back to Urbana-Champaign, joining the School of Chemical Sciences, initially as a senior research scientist, but then he joined the faculty as a research assistant professor. Before he joined the faculty at the University of Tennessee, Knoxville in 2008, where he went through the assistant professor and associate professor ranks until 2017 in biochemistry and cell biology, before he was snatched by University of Alabama at Huntsville, where he is actually a named professor and professor of biological sciences. So research-wise, obviously, so he has strong roots in MD simulations and its applications. But as I'm sure you will see in his seminar also, he moved more and more towards drug discovery, drug design, massive screening, applications of simulations and computation during years. So with that, I thank you again, Jerome, to be with us, and look forward to your seminar. Please.
Thank you very much. Thank you, Imad, for having me. It's always both, like I was telling you, a source of great pride and a great pleasure to be back in Illinois, even though it's quite virtually like the rest of our lives for this past year, essentially.
We'll get you here again. We'll get you here soon.
Yes, actually. I got my second shot 2 weeks ago, so I'm ready to party like it's 2019 now, pretty much, you know, so. All right, well, indeed, I'm going to talk about the, like Imad said, once an MD person, always an MD person. So I'm going to talk about the role of protein dynamics in the binding of ligands by proteins, in particular, in the context of conformational selection process. I'll be starting with a recap of reminding everyone what it is that we're talking about before giving actual examples of our applications. A very few words on drug discovery. About 2/3 of the drugs that you can be prescribed by a MD and that the pharmacist will prepare and give to you are actually small molecules, small organic molecules mostly, that bind to a protein and modulate its function. That's what most of the drug discovery industry is. You may not know all that, but Imad, in addition to having a PhD, is actually a doctor of pharmacy, actually. So I have to be very careful in what I'm saying today. So that's about 2/3 of the known drugs, and that's what I'm going to focus on today. About 1/3 of the drugs that can be prescribed are actually large molecules, so biologicals, essentially, like antibodies or this kind of things. Vaccines, of course, that directly perform the function. And even though my lab is involved in that as well, we actually have a narrow one on vaccine design. Today, I'm going to talk about what's at the top here. Let me, what's at the top here, which are small organic molecules binding in proteins, protein ligands, essentially. To discover a drug, the initial upstream part of the process is usually to screen the drug candidates against the protein to see if they bind. That's called target engagement. It's a very important first part. If you want to find a drug that binds to a protein, well, first you have to identify the right protein, then you express it, you purify it, you put it in test tube, and you pipette your many drug candidates and see, using biophysical approaches like NMR, mass spec, or SPR, try to measure experimentally if one of your small molecule drug candidates indeed binds to the drug. Sorry, binds to the protein. It takes time, it's expensive, 10 to 20 bucks per drug candidates, and the amount of molecules out there that the industry at large, the field could consider as a drug candidate is humongous. There are about 1 billion, slightly less than 1 billion chemicals out there that you could potentially purchase or have made for you. 230 million are routinely available in electronic formats to do molecular simulations with. So multiply 230 million by 10 bucks, and you've got a price tag of $2.3 billion at the least if you want to experimentally find drug candidates against the new protein. And I don't think anyone has ever asked the NIH for a narrow one with $2.3 billion. Although, coming to think of it, maybe it works. We should try. All right. With the FNA, I will conduct my research from my yacht in the Bahamas. If that works, I'll let you know. But most likely, you will not be able to get to $2 billion to do a test. So what we do actually is virtualize this process, right? Computational drug discovery. Almost every time a new project of drug discovery starts with actual modeling, the kind of computational molecular biophysics that you do, that we do, is actually the starting point of every modern drug discovery projects, industrial and academic. Here, in this computational process, you model the protein, you give model description of the small molecules, and you calculate the binding affinities of a chemical, right, in a protein target. And you can do it at different levels of accuracy, as you are well aware of. It can be very quick using scoring functions. It can be quite sophisticated using, say, MMPBSA approaches, or going full swing into molecular alchemy. There are different levels of accuracy and speed, but essentially you want to repeat that as much as you can to go back into the kind of high throughput screening that you can do experimentally, except that you can do it much faster and much cheaper. Okay, and that's what we do here. All right, and the idea is that you have a drug candidate. One, you compute its binding energy, and it can mean a lot of things, like a binding free energy or just an enthalpic part, depending on the level of things you are calculating. But you compute a interaction energy with between drug candidate 1 and the protein, same thing for another molecule, same thing for another molecule, same thing for n gazillion of molecules, depending on your computational power. All right. And that's what's done routinely in the field for the last 25 years. And that's what I did when I was in the pharmaceutical industry in World War I, now, I mean, some decades ago. All right. Our approach is different. It's the same principle, but we're not only focusing on one target structure, usually the crystal structure or some experimental structure, but we simulate the entire dynamics of the target structure. There is a theoretical reason for that. It's actually us trying to model the conformational selection mechanism. And the conformational selection mechanism, which I'm sure you're quite aware of, you've got a protein target and a small molecule that you would like to see maybe does bind or doesn't. In normal docking, in the docking using one structure, what most people do is they put the small molecule in the protein's binding site, and they allow some level of induced fit of the side chains and sometimes the backbone of the protein around the small molecule, and they end up with a conformation of the protein that kind of wraps itself around the small molecule, and you can create a binding energy here. What we do is actually what is observed to exist experimentally more often than not, is a conformational selection mechanism, where we have the protein and we simulate all the conformation that the protein can adopt, okay? One of these conformations or more will actually be that will accept, so to speak, that will select the small molecule to end up in the same state, final state. And then we calculate the binding energy as well. But we calculate the binding energy between this step at the bottom rather than that step at the vertical right part, okay? And there are all kind of considerations here about entropic correction coming from the dynamic, but I'll save those pleasantries for your reading pleasure of the papers we publish. But essentially that's what we do. We simulate the conformational dynamics of the target before using all those conformations to try to find what fits in there. And it's something that has been around theoretically for a while. And the first application came from your very own Robbie, in a way, when she was a postdoc with Andy. A couple of papers they published in 2008. This is one of the two ones. The other one was a more targeted application. And that's essentially that's what they do when they write. Dynamic structural information provided by MD and looking at all those conformations, and they did it on a small scale, a small number of molecules with simulation that I think were about 2 nanoseconds, so not very long, obviously. So, but it was the first time that it was described in a way that worked really. And so we've been following that essentially, but we do it in a larger scale. Well, I don't need to tell you that looking at the molecular dynamics of a protein target has a lot of consequences on what can bind. Here I'm showing a Src kinase, the surface of it, 3 different surfaces colored different ways. The blue part here, you can see a groove. That's where a co-crystallized ligand can be found, actually. There is this kind of hollow part of the surface in the green and nothing at all on the purple. If we run MD on that, you can see that the green part of the protein actually opens up and forms large cavities where small molecules could bind. And the purple one occasionally opens, like here you can see the backbone behind, so transiently open and close some potential binding site, as well as the blue one is always here. So running MD on this molecule shows that usually the binding site that you see in the crystal structures are still here, but you can have large permanent binding sites that open and that are not seen in the crystal structures. And you can have also transient short-lived binding sites where a small molecule could bind, even though the binding site is not always present. All right, so again, and I'm about to finish with the lecture part. I'm a professor after all, I like to lecture. Essentially, instead of just looking at what conformation of the protein, we enumerate all the conformations that this protein can adopt. We've got an ensemble of receptor conformations, many conformations for many proteins. We have an ensemble of potential ligands or drugs. And instead of doing the docking part on just one conformation of one protein, we look at many conformations of many proteins. And maybe we'll find that our traditional docking would identify this situation as a possible best. but we could find other conformation that could be even better. All right, and here's what we actually did about it. So because of the sheer volume of calculation, a lot, well, the MD, as you know, takes some time already. But there's a lot of conformation for a lot of proteins. And like I said, gazillions of bazillions of small molecules out there that could be of interest. We actually, to supercomputize, if that's a verb, this process, the first years of us working on that was developing parallel versions of AutodockVina, first Autodock4 and then AutodockVina, which is the one of the two docking standards. It's developed by Trott and Artelson at Scripps at UCSD. We did this development. We started it on Jaguar. We finished it on Titan. Sally Ellickson did that when she was a graduate student in my lab at UTK-RNL. She's faculty in Lexington now. And essentially, parallelizing, I'm not going to go into that, but the details, but it's actually MPII is a process of distributing the data. It's not so much the docking calculations themselves that we did parallelize, but the handling of the data, which really was a bottleneck. And parallelizing this process on Titan, for one target, we were able to use, if we had been able to use the entire machine for a day, 10 million compounds a day. We routinely did 1 million compounds a day when we had access to 1/10 of the machine, therefore extrapolate. Recently, Ada Sedova and Jeremy and their collaborators at RNL developed a GPU version. What we did was just working on CPU, a GPU version on Summit, and I'm on the paper, but it's the development part of the paper is coming from Ada essentially and her NVIDIA collaborators, where they actually went one step back in the code. Instead of using Vina, they used Autodock4, which is a version before, but much more amenable to GPU development, running on the entire machine, which we had access to. We had access to Summit entirely for a couple of days, which is really cool. It's like having a Saturn V rocket to go grocery shipping in a way. And the yield was 1 billion compound a day. All right, 1 billion with a B as in Baudry on an entire machine. To give you an idea, when I was in the pharmaceutical industry, my best machine was an Octane workstation, so that gives a bit of about my age, you know. But we had 4 cores on this powerful Octane workstation from SGI. We could have docked 10,000 compounds in about 10 years, all right, of computational time. And that was the industry, and it was a very high-tech startup. When I was in the tech, I was the tech department actually. So really, and we had a lot of success. So I want to emphasize like how totally changed is the landscape of pharmaceutical discovery in a short few years. You know, when we went to some things that like a moonshot to something that is still done on Summit, so not an easy machine to put your hands on. But in 10 years, we expect this kind of things to maybe be on the desktop of every pharmacist or every computational molecular biophysicist. All right. So we can do a heck of a lot of docking. And we've been cranking that crank happily for the last few years. And I'm not going to go into the details of all of these things, but we've been successful. However, we want to define success. We found new molecules basically that made it to the next step in virtually every medical condition and target that we have been looking at. A lot of membrane proteins, a bunch of GPCRs, so quite a diverse set of targets here. And I would like to give you 3 examples out of these many targets. The first one was done by Karen. You know Karen, he's also an analyst, when he was a PhD in my lab. And it was an interesting target. We got funded to collaborate with a pharma company, actually, through an SBIR mechanism by the NIH. And this was a protein-protein target interaction. It's called this coagulation cascade, where this protein, let's call it protein A, interacts with another protein, protein B, and we were trying to find small molecules that bind on the surface of this protein A to prevent its binding to the other partner, which is something we wanted to achieve. I'm not going to go into the biology, but What Karen did is he took the crystal structure of this protein, which he modeled in part actually, ran molecular dynamic simulation and obtained an ensemble of conformation with quite some structural diversity on the loops, as one could expect, and tried to dock a lot of molecules in a lot of different places. And what he found is that in some part of the protein, this MD simulation did change the structure quite a lot. You see the structure in yellow here, for instance, of this protein target. And you see in blue, one of the snapshots of the MD simulation, which can change quite a lot and open binding sites, like here, for instance, that could not exist in the crystal structure. You see, the small molecule that Karen did bind here in this specific example would have clashed with the atoms of the crystal structure. So MD was important to open otherwise cryptic binding site. He ran long MD simulation. He took 11 snapshots from them. He docked 200,000 compounds of each of them. We calculated that the estimated binding free energy was good for about 500 of these guys. About 100 of them were actually ordered by the company. The rest was either too expensive or impossible to find, which is a common problem. And about 24 of these 97 compounds, like not about, exactly 24 of the 100 compounds actually did bind, which is what we predict essentially in docking. We calculate binding free energies. We estimate binding free energies. So it's a 25% success rate. If you take 97 compounds and you find a quarter of them do bind, that was a big deal, a big success. And we did that in a much cheaper or faster way than screening 200,000 compounds, obviously. Out of these 24,000 compounds that bind, which is the only thing we calculate, 10 actually work, which means that they have the desired biochemical function. Sometimes compounds bind on proteins, but they don't do anything. But in order to modulate the protein, first they have to bind, all right? So here we had about 40% of biological rate and 25% of chemical success. Importantly, out of those 10 compounds that bind and made it to the next step for this company, for that company, 8 would not have been identified as docking had we done the docking only in the crystal structure. So we 300% rate increase in terms of chemical diversity, which is something we're very happy about. More recently, we did this kind of thing with COVID-19 because we all have to do something with COVID-19. And same idea, we took Oak Ridge, Nicholas Smith, Jerry Parks, and Jeremy Smith simulated, and JC was part of this project as well. They simulated the dynamics of all the targets that are part of our COVID-19's favorite virus. We looked at 3 of them, the spike, the M protein, and the M protease, and PL-Pro as well. And we docked 50,000 natural products, so compounds that are found in nature, coming from natural sources, mostly plants, but also fungi, all kind of bizarre stuff. We donated a computer, not the actual computer, unfortunately, the time of the computer. And we were able to identify, likewise, 125 natural products that seem to be binding to those 3 proteins. Then we did a bit of analyzing, like, for instance, this is a spike protein. We were targeting that part of the spike protein in blue, which is a part that interacts with the ACE2 receptor. So we were hoping to, by docking something here rather than here or there, we could hopefully prevent those 2 proteins from interacting with each other. Out of this 125, 36 could be purchased. So we sent 36 to be purchased to our level 3 collaborator lab in Memphis at UTHSC. Colin is doing that. And we have 4 hits, 4 molecules that are being now followed up in medicinal chemistry by our collaborators. All right, so 4 out of 36, not too bad, we think. At least we claim. Okay, that's an example. Something I want to. So those are 2 examples of using it and finding more molecules that you would otherwise just doing docking on the structure. I want to take a bit of a step back and look at things in the bigger picture. How important, generally speaking, is systematically is this conformational selection to identify compounds for targets. That's a work that was done mostly by Wilfredo Evangelista when he was also a PhD student in my lab. And he's back in his native Peru to be faculty. He is faculty there. So he looked at 4 GPCRs for which we have a lot of information about ligands. We know a lot using from the DUDE database essentially and its avatars. We know a lot of molecules that do bind to those GPCRs and a lot of molecules that do not bind to these GPCRs but are chemically close to the actual ligands. He worked on the adenosine 2A A2A adenosine receptor, the human one, and he ran a microsecond MD trajectory using some coarse grain. So we used a Martini force field, we modeled the structure of the GPCR in the membrane, solvation, counter-ions, 1 microsecond, and mapping back to atomistic structures to the docking. And it is the same for the adenosine receptor beta 2 and the opioid receptor kappa 1 and delta 1. Okay, so the idea is the same here. You have relatively small variations, but large enough to influence the binding of small molecules. All right. He did, out of this 1 microsecond simulation, he got 3,000 snapshots in total. And we docked the relative DUDE data sets of ligands and decoys. So about 10 million and 35 million docking calculations per protein target per snapshot. All right. So we use Summit. We use Summit way beyond our Summit allocation. And we got in a lot of trouble, but we published a nice paper out of it, and it was worth it in retrospect, I think. And here's a beat. For those 4 proteins here, I'm showing with this lovely Excel bars, a number of actual ligands that we identify through docking in 3 different ways. What if we use only the crystal structure in red, if we use a random selection of compounds, and if we use this ensemble docking, all right? For AutodockVina, for instance, out of, I think, 6,000 or 7,000 decoys that we used, we have 144 known ligands. AutodockVina loves to bind everything in its grandmother. It's a very promiscuous protein. Out of 844, if we do our docking in every snapshot, and then we select only the top 10% of the scores of the binding free energy, we identify over 800 of them. So almost all of them, all right? Even if we look at only the top 5% of the best scoring compounds, we find a little bit about over half of them, all right? Which is awesome, I think, much better than what we obtain with a crystal structure only, where it's not bad. You find about 100 ligands out of 844, but it's not as much as here. The protein docking with a crystal structure is doing barely better than a random selection of compounds, and we ran all kinds of statistical tests that I will spare you to make sure that we are showing significant things here. So for AutodockVina, running this long microsecond long MD simulation and docking in almost every snapshot, actually only 3,000 of them, led us to identify almost everything. About as well for the other targets, okay, and I'm not going to go into the details, but usually ensemble docking performs much better than crystal structure. Sometimes, interestingly, for the opioid receptor kappa 1, the crystal structure does much worse than a random selection. It's probably because the crystal structure was locked in a inactive, closed part of the structure. Therefore, the MD kind of opened the binding site there. But the bottom line is, looking at the best case scenario at the AutodockVina, we can identify about 96% of the ligands by docking in. We only need 110 conformations. In fact, we have much more than that will select a lot of compounds, but at a minimum, we found that there is this unique set of 110 specific conformations out of 3,000 that will together collectively bind 96% of the ligand, rather than about 10% looking at only a single structure. So that's wonderful, but is it good economics? It's very expensive to do all this docking, all the MD and all the docking. And so far you have to put your hands on the machine like Titan or Summit, and it's not trivial. So it still is something that is out of reach of most labs. Now it's available at your place, available at our place, you know, because we have access to this kind of machine. But getting this kind of 96% success is out of reach of most people. So the next thing we want to do is, can we predict what are those magic snapshots, as we've been calling them nonstop? We should find a better job, but a better word. But can we predict what those magic snapshots, this subset of conformation that will actually lead to the conformational selection? What do they have in common? Do they have something special? And this is an ongoing collaboration with my colleague, Professor Menon from the computer science, and our common graduate student, Shivangi. And so we're trying essentially to do super QSAR on finding what those guys have that makes them magic if we can. And so we looked at all the conformation that Willy generated and calculated a lot of physical, not a lot, about biophysical descriptor of proteins, all kind of Van der Waals surface area, eccentricity, elasticity, all kind of things. And we binned it, we cut it. It's a very complex super QSAR in multi-dimensions, way above my intelligence. Not that it's much of an achievement, but, so that's why I have to collaborate with people who know what they are doing. It's essentially a machine learning data analytics approach of trying to find what works here, using some machine learning intelligent terms that I can spell, but I couldn't do much, not that much beyond that. I know a little bit about that, but essentially this is looking at some kind of how first, it's like QSAR, it's like linear regression. Actually, those technically, they all are LR mode, linear regression. But it's quite complex because the first step is that you have to actually find the balance or imbalance in your sample, which is this part, and then do the actual modeling, which are those parts, and you're doing it in a machine learning way. So you train your own model to learn from its different modeling. All the fun of machine learning, which we are, I think, going to have to do in our field, whether or not we like it. You know, the bottom line is that applying all this semi-black magic and sacrificing goats and looking in crystal balls of machine learning, we can identify quite well, I think. We actually have, depending on the protein, between 10 and 20 times enrichment in identifying the magic snapshots that if we just select them randomly. So we have some kind of biophysical ensemble of properties that will make one of the conformation of this protein magic. It's going to be very important in the future because we cannot, like I said, screen 70 million compounds on 3,000 snapshots routine, not yet at least. So if we could find a way to reduce the dimensionality of the problem from 3,000 snapshots to maybe 10 or 20 or 50 of them, and we are able to do that, 20 times better than selecting randomly selecting randomly conformations. We may not have 96% of all the ligands, but we may go from 10% to 30 or 40 or 50%. Remember, it costs $1 billion, more than that, actually, probably $1.5 to $2 billion to discover new drugs, and it takes the best of 13 years. So even improving the process by 20% means $300 million in a couple of years, cutting down the process. So that gives you the importance of what we do, what you do. All right, and developing those tools, because it goes without saying, we use NAMD2 for our simulations and etc. So that the value of the money value and the industrial importance of that, even if it's a relative incremental improvement, translates into humongous amount of time and money. And that's pretty much it. Now, what we want to do in the future, which we're working on, is go in more details about those. Now that we know, which I'm not talking about here, we're publishing that, we know which of those many biophysical properties are often found in the magic snapshots. And we are going to try to focus on the binding site. A lot of those properties are actually large protein scale descriptors. And what happened in the binding site is not going to be influenced that much, but what happens there, but we still have to calculate everything. So our signal to noise ratio is still mediocre. We're going to focus on that. And we hope to be able to find, as much as possible, ways to predict in an ensemble of protein conformations which ones will lead to the conformational selection of their ligands. That's about it. One thing, if I may, and Imad, if I got that, I just talked about the importance of what you are doing in the pharmaceutical industry, in the science, in the research. But of course, as a Monet Kendall used to be a postdoc in my lab, I forgot to say that. He joined a few months ago. He was very successful here. We won a prize at EC20, and he's actually working in a biotech company called Polaris Biotech, where Alexander Balev used to work until a few weeks ago. He joined Black Diamond in Boston. Talk to your PI, talk to Imad, but I just heard like today that they're looking to hire someone, and I quote, who has NAMD2 and VND as their blood, so in their blood. So yeah, let Imad know if you're interested, and I reach out, and I can share the information with you, Imad, if you want. Have your PI's blessing, please. I got in trouble with RNL already. Don't get me in trouble with Imad, please. Otherwise, yeah, thank you. Well, that's pretty much it. I want to thank you. I want to thank in particular the 4 co-workers, the postdoc and graduate students in my lab who did the work I presented to you today, but all of them have been doing awesome. Thank you for paying your taxes. Please do continue. And thank you to everyone here who has given us either money or a lot of semi-stolen computational time, and we like both. And of course, thank you for your attention. Like I said, it's always a pleasure to be back virtually, even back home, back in TCBG in Illinois.
Absolutely. Well, thank you very much, Jerome, for the nice talk, nicely summarizing the state of the field, also leaving quite some time for questions. I have quite a few questions myself, but let me start with the students. Nandan, you want to start your question?
Yeah, sure. Hello, Professor Baudry.
Hi, Nandan. Good to see you.
It was very nice talk. So I have 2 questions. One is regarding that. So you used coarse graining simulation to capture different conformations of protein and selecting snapshot for ensemble docking. So my question is that as far as I know, that in coarse graining you have to use elastic network model, which restrain that you cannot capture the conformation changes of the protein. So why not then use MD simulations if you want to get diverse range of conformations of the protein?
First, we use coarse-grained simulations only on those 4 proteins that we model in the membranes. Absolutely all our other applications or projects, we use atomistic simulations using NAMD2. Here we use coarse grain using Amber and the Martini-Foss fields because it runs on GPU. So it was just a matter of on GPU only. We used Amber GPU and the Martini-Foss field. Then we mapped back to atomistic simulations. But using the coarse grain model, I'm not sure I understand your question very well, Nadan, but we did run MD simulation using coarse grain. In fact, we ran 1 microsecond for each of them.
Yes.
And, yeah, go ahead. Sorry.
Yeah. So my question is, if you use coarse graining, then you have to use elastic network model, which restricts.
Yes, that's what we did.
Conformation change of the protein, right?
Yes, that's what we did. We used an elastic network model. Yes. So
it will prevent conformational change of the protein, right?
To an extent, it does. As you can see here, I'm just showing a few out of the 3,000 conformations, but it does not prevent some variations of the loops that we found in our other projects, by the way, be it C19 or coagulation pathways, that opening and closing of the loops is where most of our new binding sites appear. You are correct. We don't want to change the folding of a GPCR, of course, but as you can see, the secondary structure is mostly preserved but there are some differences. So we do not change the global fold, nor do we want to, but we think we are able to sample the conformation well enough. And indeed, the proof is in the pudding in a way that we were able by doing that to identify many more ligands. But you're correct. Had we worked on a very large-scale conformational process, we probably coarse grain would not have been the right way to identify it.
Mm-hmm.
Yeah, I think because I never used coarse graining myself, so I was like, I don't know how much changes you can capture. But it looks like you can see some local changes even using coarse graining.
Yes, we absolutely do. Absolutely. Yeah. Enough for the docking. The docking is going to be very sensitive. Docking sorting functions are very soft, okay? It's not like the hard Van der Waals terms you have in empirical force field, like charm. So if this tyrosine is a bit further away or a bit closer away, it's not going to matter that much, all right, for a docking function. But that coupled to, you've got 6-7 angstrom differences in the location of this loop, for instance, between two snapshots. That's going to add up to have these kind of differences that we observe. So you don't need to totally change the folding. That's what I mean.
I see.
The conformation that you sample are enough for what we want to do, apparently.
Okay. So if I may ask one more question related to this similar topic. So have you considered like any sorts of approaches people have been taking for capturing, let's say, a cryptic pocket? For example, one of the approaches I know is called SWISH, SWISH, where you can change the parameter of the water so that water can interact more with the protein, and it opens up some pockets which might not be captured in the regular MD simulations. So have you thought about using that kind of approaches so that you can see not just like, let's say, stable binding, stable pockets? Maybe there is some transient pocket where you might be perform your ensemble docking approaches and might be able to identify some additional pockets.
So the short answer is no. I'm aware of those approaches. We have not used them. We have systematically used MD simulations. I want to go back on one of this thingy here. During MD, we are able to see transient pocket. You see here, for instance, this one opens very transiently. You all sometimes see the backbone, but more often than not, it's closed. So it's not only that we focus on large, always permanently binding pocket, cryptic or not, like in the green, or not cryptic like in the blue, but by docking on almost every snapshot, well, not every femtosecond, obviously, but 3,000 snapshots out of a microsecond, so I let you do the math, we are going to sample some of these binding pockets here, short, transient, cryptic pockets. Mm-hmm. But it doesn't mean that other approaches may not be able to identify them. I have not made a systematic comparison of this approach with what would be obtained from these cryptic pocket searches. That would be interesting. I agree. I have not done it. I cannot comment on it.
Mm-hmm. So Jerome, I'm going to ask you the usual question I always ask. What do you have any criterion for a conformational state to live a particular time to be considered stable enough and not completely transient? If it's too transient, of course, it doesn't live enough to bind the drug. So do you guys have thought about, I don't know, screening these snapshots based on their lifetime, to say, to filter out some of the very, very short living states that are just noise in the system and they don't live beyond picosecond to bind the drug? So do you have any sort of concern from that perspective, or is there a potential way to maybe screen these snapshots a little bit more?
Yes, this is of a very important thing, and I thank you for asking the question. We have not really looked in details either. That's something I always wanted to do, but everyone working on that has been sucked away by the pharmaceutical industry, sadly. So no, but it would be very interesting. What is the minimum time of existence for a state to be useful? Because we may find nanomolar binding affinities. If this thing exists once in a blue moon, of course, it's going to be very, very hard. But maybe not. Maybe sampling a conformation one time is enough for the conformational selection.
I see.
There's a lot of experimental results where people have measured the conformation by NMR, and not a lot, but there are some. And I know this information is out there, but no, I have not looked at it.
Okay.
It's something I would love to.
Yeah.
In fact, I would love to do some Markov state model mapping of all the conformation and their relative dynamics of existence, which also I'm not intelligent to do by myself, not intelligent enough. But that would be kind of, we would map the entire free energy hypersurface of the proteins, and we would be knowing where the magic snapshots are. We could then, in retrospect, not only identify which ones have the magic biophysical properties, but how often how much one needs to exist.
Yeah.
I don't know, and I don't think anybody else knows. So we could have a big impact here, finding that, yeah, maybe this will be a magic snapshot, but in fact, it's not going to happen. It's not going to happen because it's.
I mean, I ask especially since the agonist bound state that you guys are searching for is usually the state that is different from the apo conformation that is crystallized.
Very true.
Yeah.
Absolutely.
Yeah. Okay.
Not always. Not always.
Not always. Yeah.
Well, obviously not in 10% of the cases. All right, but most of the time, yes.
Right.
Which is a problem, more philosophical problem I have with what I'm doing in a way, is that docking calculates the interaction energy between a structure, an apostructure, and a molecule. We should include, we're going to a different hypersurface here. What we should calculate is the free energy change of the complex.
Right.
Instead of calculating the strength of the interaction between molecule and hypersurface one, which is not really relevant. It's the free energy of the complex that goes down. Yeah. And no one knows how to calculate that. So my dream, which I started to do is 10 times, but 10 times the coworkers left, is it maps the hypersurface of the apoform of the protein, maps the hypersurface of the bound form of the protein, and do we have like conical intersection or something like that between the hypersurfaces? What is the magic way to move that connects the magic snapshot on the complex hypersurface with the conformation it's coming from on the apo hypersurface? That's I'm founded a lot by collaboration with industry, and they care little about those pleasantries, but I do care a great deal. Those are excellent questions, Imad, and I hope to be able to address them.
Thank you. So
we have to convince in that case, it's not the NIH. We have to convince the NSF that this is of fundamental importance, and it is. But the NSF has not been turning my fundamental interest in this kind of thing so far.
Interesting. Yes. Maybe we can do something together on that.
I would love to.
Sure.
I would love to. Absolutely.
Kevin, you want to go next?
Yeah. So Jerome, thank you for your talk. I actually have two questions. So the first one is a general question about the field. So is it possible to predict new binding sites other than what the crystal structure gives you? Is there some systematic way to predict that? And then my second question is, how do people in this field find a balance between the lifetime of the conformation that you're selecting for and the affinity of the drug?
Thanks, Kevin. So this is a recapitulation of what we discussed with Nandan and Imad as well. So the first question, yes, like Nandan said, there are ways to look at cryptic sites, even from the crystal structures, which may be able to, and I'm sure it works well, actually. Well, Julie Mitchell has been doing that a lot at Oak Ridge, actually. Yeah, there are ways to do that. In our case, we've we've modeled the dynamics of the protein, and in retrospect, we look at where the compounds that actually work, where do they bind, and we find that, okay, for instance, that one bound in like in 80% of the case, that one bound to a cryptic site that was a dislocation, you know. So we do this in retrospect. We do our docking, we predict the molecules. When our experimental collaborators confirm that the molecule works, we go back and say, ah, ta-da, it binds there. And 8 times out of 10, it would not have been found to bind if we had used only the crystal structure. The systematic way to do that before running the MD is looking for this cryptic site the way Nandan described. Okay, and I'm not familiar with those approaches. I know they exist, and I would love also to compare those kind of things. Now, how many of those sites would have been predicted by this systematic fundamental crypto search algorithm? The second question, which is the influence of the time of existence of conformations, what we also discussed with Imad, the easiest way will be, besides calculating the Markov state surface, would just be Boltzmann weight, frankly. Boltzmann weight, the binding energy, with multiply that by the time of existence, and we may have a population. It's really interesting. It's not something I talked about here, but one thing we're particularly interested in, polypharmacology, which is a molecule interacting with more than one protein, and it's essentially system biology here. When you have more than one protein and more than one conformation of a protein, and the molecule may bind to several of these proteins or several of those conformations, you use the same kind of differential modeling that you use in system biology. It's kind of a predator-prey thing, you know. A rabbit can be eaten by a wolf and by a fox. If you have that many wolves present that often in the ecosystem versus that many foxes, which one is going to eat the rabbit? It's exactly the same thing, that if you have that many conformation that will eat your ligand, and maybe that other conformation will eat the ligand better, but it's not that present, you just do a kind of an ecosystem analysis of protein and ligands here, which is ultimately my goal, to move to a molecular information of system biology, how to use MD to feed system biology pathways modeling, which is, turn out that the error bars are so large, the noise are so large that we are far from being able to do that. And so it's like you can model anything, which is cool. But so I can't do that. But it is one a dream. As the simulations become better and the binding free energies become better, the estimate, we may be able to do these kind of things. Yeah.
Okay, thanks. So before I move to Moyon, another student, let me actually, it might be important for people to understand. So what is the state of docking in pharmaceutical industry? Is it considered a very useful and common methodology that everybody uses? You collaborate with a lot of them, Jerome. Or is it something that they use it occasionally just for the fun of it? So where are we with that?
Well, okay, so docking is vital to the process. Almost every new drug discovery project in a company, big or small, starts with docking. That is very good for my paycheck, because it's relatively easy, frankly, to find SBIR funding for companies that want to work with you if you do it. Now, MD, not so much until recently, because MD was not really useful in the process of docking. Now we think that MD is going to increase, not quite yet, because it's still very expensive to run all these MDs. But after it becomes cheaper, we think MD will be also part of it. But yes, the docking is you cannot get funded by the NIH for a drug discovery process if you do not have two things that are very important: target engagement and target validation, which means that you have to show that you're looking at the right protein target. Not only that, but the compounds you are interested in developing bind to this protein, right? I mean, as you very well know, all kind of problems of drugs being taken away from the market because we found that they bound to other proteins and
Right, absolutely.
So the target engagement is before you even do anything experimental, it will be modeled computationally by docking. What are the chances that you have good target engagement, basically?
Very nice.
So yeah, a lot of industry positions are available to you if you know how to do these kind of things. So
I hope students and postdocs are listening. So this is something you have to learn how to do if you are interested in industrial positions. Great. Moyun?
Hello, thank you for the talk. So I have two questions related to the time scale required for your docking simulations. So the first one is about the magic poses or the snapshots you mentioned. Do you find that like those magic snapshots appear like almost throughout the trajectory, your MD simulation, or they only like appear near the end, or basically what's the distribution?
That's a great question, and I know it, but I don't remember it because it's only recently that I finally could look at it. And the answer is depends. I'm going to try to find the information live if you want. The answer is it depends. Interestingly, for this kind of things, you have two scenarios. Either it's spread across, like it's the case for the Ratway, I believe, it's spread across the entire trajectory, or you have bursts of magic snapshots, like clustered together, and then relatively long period of time where the protein sample whatever it wants on the other side of the conformational galaxy, but no ligand cares for it, essentially. And sometimes, in some cases, and I believe it was occurred for the opioid kappa 1, no, for the OPRD one, you start with the right crystal structure, and actually you see the crystal structure does pretty well. And almost all the conformations are sampled in the first simulation. And then you essentially waste the rest of your simulation. As a matter of fact, we are trying to analyze that by the same machine learning approaches, where we look not at, where we introduce a time dependency on these things as well. You know, I don't know where we're going with that. I'm not even sure it makes a whole lot of sense. But yes, we do see very different dynamic behavior. And most of the time, interestingly, the magic snapshots are more or less equally, we haven't looked really at the details, but like happen during the entire trajectory. There are some cases where no, you spend a lot of time sampling the right conformation and then time sampling, wasting your cycles, essentially. If we have a way to connect not only the biophysical descriptors of the properties of the magic snapshots, but how often they happen related to time, we could really cut down on the numbers of, on the amount of computational time needed. If we cut down by, that may help us cut down 50% of the, eliminate, maybe more than that, but 60, 70% of the snapshots that have no chance of being involved in conformational selection. If we can also increase that 2 or 3 times by looking at some time dependency, we may end up being able to eliminate 80% of the snapshots, which would be a great deal of time saved here.
Thank you, Jerome. So let me actually end. I don't see any other questions. Do you have other questions?
Yes, I actually.
I'm sorry. Go ahead.
I think you already answered my second question. Is like how many like snapshots do you need to really achieve like good ensemble docking result? I think you already like answered that somehow, but a short follow-up question is about the machine learning work you are currently working on. So do you think those knowledge learned from this machine learning technique can be like very generalized, or that still depends on the system?
That's a very good question. And we're looking at a great deal of details of what physical descriptors are common across those proteins and common across those different linear regression smooth approaches. It's a bloody chaos at this point. Nice. But we do find that there are some descriptors that are found in most of them, virtually, in almost all the statistics on 3 points so far, but in every case, okay? But not very many. And they correspond nonetheless to about half of the enrichment. So think in terms of PCA space, there are a small number of descriptors that are responsible for about half of what we see being important, which means that there are about another 10 or 20 descriptors that are specific to each protein, we think. All right, so it's going to be a mix of, it's going to be a mix of things that are very, very universal, but maybe obvious, you know, like radius of gyration of the binding site. We may find out that, yeah, the more the binding site is open, the more you can bind something in there, which is kind of trivial. And maybe, and things that will be specific to every binding site. We're not there yet, but we hope to be able to rationalize by binding site class, by something that we're looking for. We are looking for a target where to send our arrows now here. Here's a good, if I may end up on that, the difference, I learned that many times, the difference between academic drug discovery and industrial drug discovery. In industrial drug discovery, you have a target and your goal is to send the arrow at the center of the target. In academic discovery, you shot the target in the air, it falls on the ground, and you go there and you paint a target around it. So I mean, I'm in the latter here. I'm trying to find what is it good for, kind of.
Well, thank you very much, Jerome.
Thank you for your
That's
answer.
really good. So I think we should maybe touch base. And I was very glad that you pointed out this might be a way for MD to become more important for pharmaceutical industry. That's a very important point you make. And so we should push on that front a little bit more to make it even more interesting for people. So if there is an opportunity to interact and collaborate without stepping on somebody's toe, it would be
those are my toes, and I would love to step on them, believe me.
Okay.
And with your permission, Imad, let me remind everyone that if you want to hear about this drug discovery company that is specifically looking for someone who has NAMD2 and VND in their blood, let me know.
Oh, absolutely, absolutely, by all means. Okay, so I know that you're meeting with a couple of students after this. Other people, if they are interested, are more than welcome to stay around.